Hi, biologists. Happy Monday. I hope you guys had a good weekend. Great to have you back. And let's get started for the week. So today is Monday, April 6th. So what we're going to be working on today is our same standards. So we're talking about how traits are being passed down, and then our objective for today is to determine what codominance and incomplete dominance are by completing practice problems. You guys are going to have a little practice problems with those and then analysis. So as always, there's my contact information. So let's get started. So talking about dominance, we can be talking about different types of dominance. So when the dominant allele shows up in an offspring and it automatically covers up the recessive allele, meaning the recessive allele isn't showed at all, that is known as complete dominance because the dominant allele is taking over and the only phenotype being expressed. There's also something known as incomplete dominance. So this is in heterozygous individuals. And so this is when they're blended together. And I believe I misspoke from the last video and mixed up incomplete dominance versus codominance. So incomplete dominance is when the alleles are blended together. So if we have different colors, if we have blue and white as our dominant versus recessive allele, and then we have a heterozygous individual, it would express both those alleles. So you would end up with a lighter blue when you have blue and white, for example. And then codominance is when in heterozygous individuals we see phenomena like stripes and spots. So both alleles again are getting expressed, and it's resulting in this kind of more non-solid patterns. So if you guys are on the first page of your packet, so I have everything that we just discussed listed above for your convenience. And then right now, let's complete 1 through 3 of the on page 1. So determining what complete dominance, incomplete dominance, and codominance would look like in these specific examples based on what you just read. So you can use this, for example. So let's complete 1 through 3. So pause me now to do that. Okay, welcome back. So by now you should have completed 1 through 3 of on page 1 of your packet. So let's go over them really quickly. So I'll give my annotation, what color, let's use red today. Okay, so complete dominance. So if we have complete dominance in this first one, so what would our phenotype be looking like if we have a RR individual? So big R, little r. So complete dominance means that the dominant allele is completely taking over. So if we have complete dominance, we would see a red flower because this dominant allele is completely taking over and not even letting the recessive allele of the RR be expressed. Incomplete dominance, so if we have, again, this RR, big R, little r, and incomplete dominance. So incomplete dominance means that they're blending together. So if it's a red flower and a white flower, we would have a pink flower because it's a blending together for incomplete dominance. Okay, and then finally, so if we have codominance, so remember codominance, we have stripes or spots. So if we have this homozygous individual or heterozygous individual for codominance, then we would have like a red flower with white spots or whatever, or stripes. Okay, so pause me now if you need to write this down. If you have any questions about that, please shoot me an email or write in the comments. So we're going to move on. So on this next page, on page 2, we you guys are having a lot of practice problems today. So a lot of practice. So I just want to quickly go over that. So for questions 4 through 6, you're going to be learning and discussing snapdragons. So they're a type of flower. I wanted to give some context. So here's what the flower looks like. So snapdragons are incompletely dominant for color. So remember, incompletely dominant means that they blend together their colors when they have a heterozygous individual. So as a result of this, they have phenotypes of red, pink, which is the blended color, and then white. So of course, the red colors are these homozygous dominant. White flowers are homozygous recessive, whereas these pink flowers are heterozygous. So, based on this information, let's complete number 4 through 6, which is up until this bottom part here. All that I've highlighted for you. So let's do that. Pause me now to do that, and then we'll go over it, and then you guys will have more time, and then we'll go over it tomorrow in our live session. Welcome back. So by now we should have completed our so we should include 4 through 6 on page 2 of our packet for today. So let's go over it. So a red snapdragon has the genotype of homozygous dominant. And so you guys kind of get, and it tells us here that it wants us to use big R and little r for our list. So a red snapdragon can only be big R because this species of flower exhibits codominance or incomplete dominance. I'm sorry. Incomplete dominance, meaning that the only red flower is going to be the homozygous dominant. So pink snapdragon is our heterozygous individual because it's the result of the blending of the color. So it's big R, little r. And then finally, for the white snapdragon, it's 2 little r's because it's homozygous recessive. So then it asks us to do a Punnett square below for a pink flower. So we know that our pink flower is going to be this homozygous recessive. So one parent is going to have RR, and the other parent is going to have RR as well. So then you can do the cross real quick. As you see that R goes to R, R, and then the same thing. Okay, so red we see is gonna be large R, large R. So we're gonna put 2 big R's up here. White is 2 lowercase r's. So they all are going to have the same exact phenotype because each parent can only donate one type of allele because they're both homozygous. So we have 1 large R for each of these possible offspring, and then a lowercase r as well. So pink and white. So pink is heterozygous, whereas white is homozygous recessive. So this is what our cross will look like. Okay, very good. So we get half of our cross expressing the pink gene or the pink phenotype, whereas half is expressing the white phenotype. So it asks us about genotypic percentage. So this means what are the different genotypes and how, like, what's the different percentages that we're showing here. So for, oh my goodness, my kitty cat is all up in my business this morning. So genotype, so we're going to be seeing 1 out of our 4 are homozygous dominant. So I'm gonna just put D because I don't have a lot of space, so I apologize for that. Hopefully you guys have a little more space. 2 out of the 4, so 1/2 is heterozygous, and then the other 1/4 is recessive. So for this next one, 4 out of our 4 are heterozygous because they're all heterozygous. So they're all 4 out of our 4 are going to be pink. Okay, and then sorry, we can go back to the first one over here. So 1/4 are dominant, meaning 1/4 is going to be red, 1/4 is white, and then 1/2 is pink. Okay, so for this one, we have half of our offspring are heterozygous, and then half are recessive, homozygous recessive. So this H here, so H for me is heterozygous, and then R is recessive, and D is dominant. So homozygous dominant, homozygous recessive. So that means half, if we have half heterozygous, so that means half are going to be pink, and then half are going to be white. Okay, so pause this if you need it. If you have any questions, again, shoot me an email or put them in the comments. I want to clear that so we can move on. So now that you guys got a hang of it, we're just kind of going to breeze through the rest of it. So pause me now to complete 7 through 9. So this bottom half of page 2 of your packet. So pause me now to do that. It's basically asking similar, exactly the same thing. It's just talking about horses now that are experiencing this incomplete dominance instead of the flowers. Okay, so welcome back. So you should have completed up until the bottom of page 2. So these Punnett squares on horses. So now we're going to move on to these questions 11 and 10 that are dealing with the horses as well. So the little analysis questions. So thinking about, can these palominos be considered purebred line of horses? And then what 2 colors of horses would you want to breed to produce the largest amount of these palominos in the shortest amount of time? So pause me now to do that. And again, we're not going over these today because we're going to go over them in the live classroom, and I'll post the video for that onto the YouTube. So pause me now to complete these page 11, numbers 11 and 10 on the page 3 of your packet. Okay, welcome back. So now you should have completed 10 and 11 on the 3rd page of your packet. So now let's complete 12 through 15 on the page 3 of our packet. So you are just going to complete questions about these weird regular circular-eyed versus starry-eyed versus circle star-eyed individuals here. So pause me now to do that. Welcome back. So you should have completed numbers 12 through 15 on page 3 of your packet. So good job. Again, we are going to be going over these in office hours basically on tomorrow, on Tuesday, April 7th. And I'm also going to post the answers to these on Google Classroom at the end of the week. So on Friday, April 10th. So now we're going to go into what is called codominance. So remember, codominance is when both alleles in a heterozygous individual, both the dominant and recessive allele are being expressed, resulting in something like stripes or spots. So blood type is an example of codominance. So we all have blood. So blood type is another thing that you get from your parents. So I don't, if anybody's ever gone to donate blood or needed to receive blood, this should hopefully sound familiar. So on your packet and on the screen here, I have a chart that gives us our blood type. So that's the phenotype. So if you have A blood, I have A positive blood. You guys might know your blood type, so this might sound familiar. So these are the different blood types that are possible. Genotype is here. So genotype is shown here. If you guys note up at the top, so blood type exhibits codominance, the two different dominant alleles are A and B, whereas the recessive allele is O. So the genotype here follows typical genotypic nomenclature. So O is represented with two lowercase i's. So because it's recessive, so they're lowercase i's. So that's it's a little different than what we're normally used to, but it still follows the same conventions of the lowercase versus the uppercase for the dominant or recessive versus dominant alleles. Since both A and B are dominant alleles, they both have the uppercase I. They're just represented with little superscripts of A versus B. So AB blood type would have both A and B alleles. So they're both uppercase I's because they're both dominant, and then they just have their little superscripts up here. And then, so A can also be expressed with one, just again, because it's dominant, it can have one of these uppercase dominant alleles, and then a recessive allele, the lowercase I, and same thing with B. And then over here, it tells us which blood types can donate which type of blood to which individuals, and then which blood types can receive blood from which type of individuals. So here we have O being the universal donor because it has this recessive allele that can be donated to any of these. Whereas AB is a universal receiver because it has both the A and the B dominant alleles, and then it can also receive the recessive allele from type O. So now you guys are going to complete the, let's do numbers 1 through 3 on page 4 of your packet. So right here. So 1 is asking you to basically just look at the charts and answer these questions here. You have everything you need in this chart for 1. And then 2 and 3 are asking you to do crosses, and we'll go over those together. We won't go over 1 together because it's basically just going over the chart. We'll go over it in the live classroom tomorrow, and then I'll post these answers on the packet at the end of the week. So we'll go over 2 and 3 real quick. So pause me now to do that, and then we'll go over it together. Okay, so welcome back. You should have completed 1 through 3 on page 4 of your packet by now. So we're gonna quickly go over one of these. Let's do number 2. Let me just get my writing utensil out. So number 2 says, pretend that Jay-Z is homozygous for the type B allele and Beyonce is type O. What are all the possible types for their baby? So if we look up the type B allele is he's homozygous. So remember, Jay-Z is homozygous for this B allele. So we see that the B allele for homozygous is only this uppercase I with the superscript B, uppercase I, superscript B. And then Beyonce is type O. So the only possible genotype for O is up here. So lowercase I, lowercase I. So they both are homozygous for their traits, so their child may only have this specific type of blood. So their child there's a 4 out of 4 chance that their child will be heterozygous for type B blood. Okay, so you guys will do the same thing for 3. So we'll go over that tomorrow in our office hours. So you guys, I know it's a lot, but let's just complete the rest of the packet. So pause now and then we'll end. So complete the rest of the packet, pages 5 through 8. So it's just more practice with blood type and codominance. And it's also really interesting. So seeing who can be potential parents to different offspring. So pause me now to complete pages 5 through 8. Okay, welcome back. So you should have completed pages 5 through 8 of your packet. Good job. I know it's a lot, just a lot of different practice problems. So really good job. You completed the entire packet for Monday, April 6th. So good job. You're a third of the way through the week. So just a little look ahead to what we're doing. So we're going to have our live classroom tomorrow, 4 through 7, where we're going to, for April 7th, 9:30 to 11:20, where we're going to go over all these practice problems together. We have another video upload on Wednesday, the 8th, another live classroom, and then another video upload. So as always, here's my contact information. Please reach out to me, text, call. I try to get back to you as quick as I can. See you guys next time.